All right, welcome back. We are continuing our discussion on gene expression and protein synthesis. So this little mini video, mini lesson is just going to do an overview before we jump into the processes of transcription and translation. So I'm going to try to give you the big picture first, so then you kind of know where to place these concepts once we get into a little bit more of the nitty-gritty details of transcription, translation, and what can happen when things go wrong. So I'm calling this gene expression overview. So a kind of a pre-summary, if you will, of what we're going to be covering. So this is my overview slide. So here is the idea. Remember our central dogma, DNA to RNA to protein. Right? And so that's what we're seeing here in this picture. So the blue double helix ribbon, that's our DNA. It holds the gene. So let's say in this particular picture, this is the gene. Just that short little sequence of the DNA holds the A's, T's, C's, and G's, all the instructions for the protein of interest. If you want to stick with hemoglobin, we'll stick with hemoglobin. So let's say from here to here, that is the instructions, the A's, T's, C's, and G's in a very specific sequence in that right order to code for the protein of hemoglobin. Okay, so that's just the DNA. The transcription is that process of taking the language of DNA and turning it into the language of RNA. And RNA language is a temporary copy. It's not a permanent copy. I have an analogy I'll talk about once I go through this overview. So the transcription is a temporary copy, and you still have the base pair ruling. So wherever you have a T, right? So if this is a T on the DNA, it's going to match up with an A in the newly made RNA. If it is a C, you're going to have a G. But here's where that we had those two, the new base, the uracil. So if your DNA codes for an A, like we see here, instead of a T on the RNA, it's going to be this U. Okay, but uracils are molecularly close to thymines, and so it's going to make a good match. So you're going to have those base pair rules. So just like in DNA replication, whatever you know one side of the DNA, you're going to know your complementary RNA because of those base pair rules. You just got to substitute uracil for thymine. So then in the process of transcription, you're going to make this temporary copy of this DNA bases into your piece of RNA, and we call this mRNA. The M stands for messenger. So it is the messenger of the instructions in the DNA to take it out to the ribosomes. So if you remember your cellular structure, ribosomes and their function is protein synthesis. This is the site of protein synthesis. So that's where we're going to be seeing those ribosomes really do their work. So the mRNA goes and lands on a ribosome, which is not pictured here. That's more of the details we'll get to later. And it starts reading these instructions 3 bases at a time. 3. So this first base, AUG, this is what we call a codon. A codon is the 3 bases of mRNA that codes for a single amino acid. So in this case, the sequence AUG specifically codes for the amino acid methionine, and that's going to be the first amino acid in this particular protein. The ribosome will then move on to the next one, ACU, and ACU is going to code for the amino acid threonine. And the ribosome just keeps on plugging along, reading each codon. So methionine, threonine, cysteine, glutamic acid, cysteine, phenylalanine, until it's done. It's just, this is the done end, right? It's a little bit shorter than it should be just for the purposes of this picture, but it will continue all the way down the length of the mRNA until your protein product is done. And then you will have your primary sequence of your amino acids, which will then fold secondary, tertiary, and quaternary if that's part of its structure. Okay, so this is our central dogma, DNA to RNA to proteins. And so the processes of transcription and translation are how that central dogma works. So transcription copies DNA to RNA, and translation converts RNA to the amino acid sequence. Okay? So here's my analogy. So a lot of times people consider the DNA is the blueprint or the master plan for the cell. So I like to build on that saying and say, okay, So if you're at a construction site that would actually be needing the blueprint, where would the master plan, right? So you're on a building site, you're building the new science building up in Grants Pass, or the nursing building that's going out in White City, whatever, you're building a new building. Your master plan is going to have all of the instructions for every piece of that building, for the electrical and the plumbing and the finish work and the tile and the floor and the carpet and the windows and everything is all going to be housed in the master plan. Now, is that master plan something you want to take out on the work site? The whole master plan if all you're going to be doing is windows? Well, no, usually the master plan stays in the foreman's trailer or the architect's office or whatever, and the master plan stays protected. But if your job is to do a particular piece of that building, all you need is the page or pages that's going to give you the instructions for your particular job. So you can think of the DNA as the master blueprint, right? It has all the instructions for you as the product, as the person, but it stays protected inside the nucleus. And every cell has the entire copy of your entire blueprint. So even your skin cells have all the DNA, all 46 chromosomes in every single one of your skin cells has the instructions for your liver and your brain and your kidneys and your bones and your muscles. But in your skin cell, all it uses are the pages that are associated with being a skin cell. Your liver cell has all of the instructions for every cell in your body, but it only needs the pages to allow it to function as a liver cell. So the master blueprint stays in the nucleus, very well protected. The mRNA is just the pages of interest. What are you doing at this moment? What is your instructions? And then the protein is the product. All right. Are you putting in a toilet? Are you putting in a window? Are you running the electrical wire? Are you laying down the floor? You just need the instructions for that one thing that you're doing. So your DNA is the master plan. It's got all of the instructions for everything. Your mRNA is just a copy of that one instructions, and then your protein is the product of those instructions. Now you might be asking yourself, as I was going through this earlier, how does the ribosome know that UUC, those three bases, code for that particular amino acid? This is called phenylalanine. How does it know? Well, a lot of work went into trying to solve that mystery, and researchers came up with something called the genetic code. Sometimes it's called the universal genetic code because this genetic code is true for every single living thing. And viruses. Viruses are not living, but they still follow this genetic code. So viruses, bacteria, archaea, fungi, protists, plankton, single-celled organisms, which are protists, animals, plants, I already mentioned fungi. Every living thing that we know that has ever existed, pretty much, uses this genetic code. So scientists, once we were learning about DNA and we learned about this process of transcription and translation, they use their super smarts and technology to figure out, elucidate, it's what it's called, to actually solve this mystery of what these three-letter codes code for in the particular amino acids. So if you were to go back to chapter 3, you can if you want to, but you can flip through chapter 3 when we're talking about proteins and the macromolecules. There was that kind of big fold out pages of all of the amino acids, and I said you don't have to learn that, but if you're interested, those are the amino acids. Right, I had the amine group on one side, a couple carbons and a carboxylic acid group, and then the R group was different between each amino acid. So there are 20 amino acids. Okay, our bodies, our entire body, for all the proteins that we need is just a combination of those 20 amino acids in various sequences, and the various sequences allow it to fold into that proper shape to do its proper function. Now, when we take a look at all the possibilities of a three-letter code using just four letters, we can do a little bit of math, and what we end up with is 64 possibilities. So we have more spots, if you will, than we have need for the 20 amino acids. So there's actually a little bit of a redundancy. So what that means is if we take a look at proline right here in the middle, proline actually has 4 different codons that code for proline: CCU, CCC, CCA, and CCG. So there's a little bit of redundancy, and we're going to talk about that as we move along. There are some that only have 1, like this AUG only has 1 3-letter code. So the only way you're going to get methionine is if you use the letters AUG. And there's another one called tryptophan. Yeah, it's the same tryptophan that's in turkeys that make you sleepy after Thanksgiving. Its instructions is UGG. So those are the 2 amino acids that only have 1 codon code for their particular position in a protein. All the other ones have at least 2, and some of them actually have, I want to say some have 6. Yeah. So leucine actually has 6 different codes that when the ribosome reads any of those 6 codes, the amino acid leucine will come in. And you can see some of the other ones. So, okay, all right, so they've figured that out. So how does the ribosome know what to do? And this was always my favorite part when I was learning this whole process, because it's kind of like a mystery. It's kind of like, it's like a decoding ring. Can you solve the mystery of this secret language? So let's say I have mRNA that has been read off of the DNA in the process of transcription, and it's got this sequence. And I just kind of, those letters off the top of my head. So usually what I like to do if I'm trying to translate kind of on paper, not in the actual picture with the ribosome, what I'm trying to do is figure out what amino acids are going to be coded for by this sequence of letters. Well, every 3 letters codes for amino acids. So let me underline the 3 letters so I don't get mixed up. Okay, so it looks like I should end up with 4 amino acids in my little short polypeptide. So the way that this works, I'll try to get my highlighter out here. So the way that the genetic code table works is on the far left-hand side, you're going to be looking for what is the first base on the 5' end of the codon. Remember, codon is RNA, so it's going to have the 5' end. So my first letter is A. Okay, so I'm going to find my A over here. And so everything in this box going horizontally has A as the first letter in the code. All right, so then we take a look at the second letter is U. So now I find U up here in the second mRNA base. And so now everything in this box has U in the second base position. So it's almost like a multiplication table. So we've highlighted this box right here. So this box has AU. All of the codons have AU. And then if we take a look at the last letter, G, then we come over here to this part of the table and we find G. I'm going to get a different color because I've got too much yellow going on. So then we go across this way and every codon has G at the end. So that gives us our final answer, AUG. So AUG is going to code for methionine, which just happens to be the start codon. So every protein starts with methionine, which is so amazing. Well, let's take a look at the second one. Okay, first letter is C. Let me erase all this stuff. There we go. So if we take a look, our first letter is C, right? So now we're going to be looking in this box here because the first letter is C. Then we have G. So now we're going to be looking over here. I'm just going to circle that one box because that's all we need, C, G, G. So then our third codon over here, C, G, G, is going to code for arginine. Okay, so you kind of get it? See if you can do the next one. I'll give you a minute to kind of walk through that as I erase my things. So A, U, U. Okay, so A, U, U, right here. So that's going to be isoleucine. That's ILE is what that, oops, isoleucine. Okay, one more. So I'll give you a minute to see if you can get UGA while I do my erasing here. UGA. Okay, so now we're up here, U in this box, G is in this box, and A is right here. So this is a stop codon. Hey, there's built-in instructions to tell the ribosome we're done. So in this particular example, we have a really short, just a tripeptide methionine arginine isoleucine, and that's as far as I was able to go. But you know, proteins are going to be hundreds of amino acids long, so that's going to be times 3 bases long. So this is your genetic code. So it's really important that these sequences start in the right starting point and that there's no mistakes, right? So we had the example of the hemoglobin with that one letter mistake can completely change the outcome of the protein. So the last slide is kind of illustrating this idea of what we call reading frames. So the reading frame is a way to make sure that the amino acid starts properly. Now I'm just going to erase, just ignore this right here. I just want it to start on methionine, so forget that that's even there. So we're going to start at AUG. So AUG is our start codon. So AUG, UUU, AGC, GCA, AUC, AGG, AAG, UGU. This is normal. Oops. Normal. That's going to give us the proper sequence of those amino acids in the green letters to give us our functional protein. Well, take a look at what happens if we, again, we're ignoring the first 3 bases. So ignore that. So what if we start one letter down? Right? So instead of having AUG, which is going to give us methionine, what if we go UGU and then UUA? We're not changing, we're not mutating, right? We're not changing any of the letters. We're just changing where it starts from. How is our sequence compared to the normal? So the normal is methionine, phenylalanine, serine, alanine, isoleucine, arginine, lysine, cysteine. Our new sequence, if we just shift over one, cysteine, leucine, alanine, glutamine, serine, glycine, serine, got to remember my amino acids by their abbreviations, is completely different. There's no way, if that red sequence of amino acids are made from that RNA, mRNA, there's no way that's going to fold to have any resemblance of a normal functioning protein. Well, what if we start down here? And erase those. What if we start two down? So instead of AUG, we're going to go GUU. So we're going to get a valine, right? And then what do we get? UAG. If you were to look in your genetic code that you have in your textbook, I can't flip back to the one on the screen, but it's a stop codon. So you'll have one amino acid in your protein, and then none of this would continue because the ribosome says, oh, stop, ping, it's going to come apart, and then you're going to be done. So this is what we call the genetic code. And the reading frames are super duper important to maintain. So this is a mutation, and this is a mutation. And we'll see these different types of mutations. So this is bad, right? So this is going to be a non-functional or even a non-existent protein, all right? So that is kind of this introduction to the overview of protein synthesis, with the DNA is the master plan. The RNA is just the page for that one gene that codes for a particular protein. And the protein is the product, it's the end result. And then we use that genetic code to determine, the ribosome uses that genetic code to read the codons to know which amino acid gets put in the right place. So the next couple videos are going to be on the specifics of those processes, transcription first, and then we'll wrap up with translation, and then a video on mutations and some other topics. All right, I will see you next time.